This is a chapter on the cellular elements of the brain, the neurons and neuroglia. The authors of this chapter are Carl Harrop, Kai Hetze, and Bruce Trapp. The key concepts that are presented in this chapter are quite simple to summarize here. Most important is that the cells of the brain are very diverse, both in their structure and in their function. Despite this diversity, the cells must cooperate intimately for the brain to function properly. All cells must cooperate, and in doing so, all cells contribute to cognition. Neurons transmit the electrical signals from one place to another in the brain. Oligodendrocytes determine the speed of transmission. Astrocytes help maintain metabolic homeostasis. Microglia prune synapses and have other functions as the innate immune system. And the vascular endothelial cells help to maintain the metabolic balance in the brain. The fourth point is that while most cells in the brain are mitotically inactive, it's important throughout the life that the cell cycle activity of all the cells be suppressed. This is a way of guarding against tumors and protecting against cell death. And finally, and most recently, molecular techniques now allow us to describe an extraordinarily high-resolution molecular signature at the level of a single cell. Let's start with an overview of the basic components, and let's start with the neuron. Neuron has three main parts: the dendrites, which are like the antenna of the cell; the cell body with its nucleus that serves as the anchor point; a specialized region known as the axon hillock, where a single fiber known as the axon leaves the cell body to take the electrical information from one cell to the next. The little sausages that you see along the axon, we'll learn later, are actually pieces of myelin. The signal transmission is along the axon towards the next cell, and when it reaches the next cell, it ends in a specialized ending known as a synapse, typically on another neuron, and we'll see more about that in detail. The structure of the neuron can be extremely diverse. These three cells are all considered neurons, and yet you can see how different they are in size and in shape. The one on the left with the beautiful fan dendrite is a Purkinje cell, and you can see its dendrites displayed off to the left, the cell body down towards the bottom right, and a single small fiber, the axon, coming off at the lower right. That axon, like all other axons, will end at a synapse, and that's diagrammed here, where the electrical signal from the axon is transmitted to the next cell through a chemical synapse, by which the electrical signal is turned into a chemical excretion, followed by return to electrical activity in the postsynaptic cell. Let's look at the cell body and the various ways in which we can view it. This is an example of two Purkinje cells sitting right next to each other. They've been stained with a purple dye. You can see the cell body in its central nucleus. It's also possible to look using a technique known as Golgi impregnation, where silver salts are deposited. This time it fills every single process of the cell, its dendrites, although not its axons, but it gives a very, very different picture of the cell. It's also possible to look at messenger RNA within the cell, and that technique is known as in situ hybridization. These three purple blobs are also Purkinje cells, but they've been hybridized with a probe specific for a messenger RNA that is unique to Purkinje cells. This picture is a picture of an electron micrograph, where a very thin section is taken through a cell just like the ones on the left, but it's now examined in an electron microscope. And finally, you can use antibodies to do a technique called immunocytochemistry, which also reveals the cell body of a neuron. The green here is an antibody that stains the dendritic and processes of the cell and the cell body. And the tiny little red line indicated by the arrow is a specialized part of the axon known as the axon initial segment. That initial segment is actually worth looking at in greater detail because it is the go-no-go decision point for whether or not the neuron fires an action potential. The structure has been worked out most elegantly, and it can be seen to be a series of transverse actin bands surrounded by high densities of sodium and potassium channels that allow the initiation of the axon, of the action potential. No neuron functions by itself, and in fact, neurons function in circuits, and that is where the subtlety of brain activity actually comes from. Circuits can involve both excitatory and inhibitory links, and the circuits cannot function with neurons alone. All of the circuits need neuroglia, and that brings us to the second part of the chapter, which talks about the second type of cell in the brain, namely the neuroglia. Neuroglia are believed originally discovered as the support cells of the brain. They help maintain neuronal structure and metabolic function and are essential to maintaining proper neuronal activity. There are many subtypes that can be broken out from neuroglia. They include astrocytes, which we'll learn about and we'll learn about these in order: microglia, ependymal cells, oligodendrocytes, and their precursor cells, the OPC cells, as well as the Schwann cells, peripheral nervous system. These are the astrocytes of the brain. On the left, you see an immunocytochemistry image of an astrocyte stained in green, and the red is a blood vessel anastomosing through the brain, and you can we pick this image to show you the intimate relationship between the two. The diagram on the right is a more easily digestible way of looking at how the astrocytes interacts with the other cells. It surrounds the blood vessel and interfaces at the surface of the brain, known as the pia, and in so doing helps form the blood-brain barrier. The astrocyte also connects directly to the neurons and to their synapses and to the space between two myelin sausages, and in doing so helps provide metabolic support to the neuron and to the other neuroglia. It also has a role in inflammatory responses and is known to secrete cytokines during an inflammatory process. The real cell, though, of the innate immune system is known as the microglial cell. These are the equivalent of peripheral macrophages, and they serve behind the blood-brain barrier as the brain's innate immune system. In addition to that, they're incredibly important during brain development, and they're required to maintain the integrity and the health of synapses throughout the brain and into adult life. They are phagocytic cells and help clear debris, and one classic example of how they do that is where they are asked to clear the amyloid plaque created during the course of aging and Alzheimer's disease. And you can see the red microglia trying to eat the amyloid plaque by phagocytosis. Next is the oligodendrocytes and their precursor cells, the OPCs. Oligodendrocytes are found only in the central nervous system, where they synthesize myelin, the sausages along the axon that help speed up and make more efficient the transmission of the action potential. The rate of myelin synthesis changes with neuronal activity, so there's a very strong feedback relationship between the neuron and the oligodendrocyte. The oligodendrocyte precursor cells sit and wait for when they are needed to replace dead or damaged mature oligodendrocytes, and probably serve other functions in the brain as well. In addition to the well-known support of neuronal transmission velocity, they also provide trophic support to neurons and help maintain their metabolism. The Schwann cell is another type of neuroglial cell, It is found in the peripheral nervous system. Functionally, it works exactly like the oligodendrocytes of the central nervous system, but the two cell types never intermingle. Unlike the oligodendrocytes, which are permanently postmitotic once they've begun the process of myelination, the Schwann cells remain mitotically active and can dedifferentiate and re-enter the cell cycle to multiply and help remyelination when it is required. The final part of the chapter is really a work in progress, but a very exciting one. Using molecular techniques that have been reduced to nanoscale, we are now able to form libraries of the messenger RNAs in single cells. These can be amplified, sequenced, and when that's done for thousands of cells, we get expression profiles from an entire range of different cell types. These can then be statistically separated into what are called clusters, and while they're displayed in 2 dimensions, what's really going on here is they're clustered statistically in a multidimensional space that's impossible to render in 2 or even 3 dimensions. Based on the different expressions of different messenger RNAs, we can identify parts of these clusters as being specific for oligodendrocytes. As you can see in the upper right, the cluster that is blue in panel A is now red. That's expressing the oligodendrocyte-specific protein MOG. Excitatory neurons spread over more clusters, but OPCs are, as you can see, situated primarily in a single cluster that sits right next to the oligodendrocyte cluster. This technology is only now beginning to be exploited, but it will be a new and exciting way of looking at the brain. And that is the essence of the chapter. To conclude our major points, our brain cells have incredibly complex structure and chemistry. They have evolved to function not as individuals or even as individual cell types, but rather as an ensemble of cells. When their functions are combined, they produce networks, and these networks have extraordinarily complex computational power, which gives our behavior and our cognition the subtlety that we all have come to expect from it. But the cost of this high level of function is that the absence or malfunction of any cell, be it neuron or neuroglia, degrades the network and interferes with cognition. And the authors wish to thank for her help in assembling the graphics that went with this chapter, Ms. Candace Kent. The financial support is listed here, and these are the affiliations of the authors of this chapter.